Now, an ancient man is revealing that the secrets of Egypt, the country where the history is full of mysteries.
From pyramids to pharaohs, every artifact has fascinated us all. Scientists were stunned when they opened a sealed clay jar. Now, this jar was hidden deep within a tomb carved into an Egyptian hillside.
In 2024, scientists accomplished what was thought impossible. They cracked the genetic code of Egypt's oldest tombs. The answer upends everything. These remains from 5,000 years ago reveal early Egyptians were not isolated, but a striking blend of local North African and ancient Near Eastern ancestry. There was no foreign elite. Instead, a centuries-long fusion rewrites the origins of Egypt's earliest pharaohs. If history's most iconic civilization emerged from unexpected roots, what else about ancient Egypt have we misunderstood? 5,000 years ago, the banks of the Nile stretched wide and green, sheltering the first tombs of a civilization on the rise. In Upper Egypt, near the village of Nuweirat, a hillside was carved open to make space for the dead. Here, the rock-cut tombs held more than just bones. They preserved the silent beginnings of Egypt's story. One burial, sealed inside a thick-walled ceramic pot, lay tucked within limestone, protected from the searing heat and shifting sands above. The pot itself was large enough to cradle an adult in a flexed position, a tradition that spoke to both practicality and belief. Layers of Nile silt and sand insulated the chamber, creating a microclimate that buffered the remains from the extremes of the desert. Temperatures inside these tombs hovered between 20 and 28 degrees Celsius, with humidity rarely rising above 60%. These stable conditions, combined with the natural shielding of the ceramic vessels, slowed decay and preserved tiny fragments of ancient DNA. Not far to the north, the sprawling necropolis of Saqqara marked the edge of Lower Egypt. Here, tombs took the form of mastabas, low, flat-roofed structures built from mud brick and limestone. Underground, burial shafts reached depths of 5 to 10 meters, their chambers sealed with stone and filled with sand. The deepest rooms, cut into the limestone bedrock, stayed cool and dry, maintaining temperatures as low as 18 degrees Celsius and humidity below 45%. Some burials at Saqqara also used pottery coffins, echoing the traditions seen further south. The sealed chambers and alkaline soils offered rare protection, allowing bone and tooth to survive for millennia. At Abydos, in the desert west of the Nile, the tombs of the earliest kings rose from the sand. Mud brick enclosures and deep burial pits shielded the dead from the harshest elements. Here, bodies were wrapped in linen or mats, placed in wooden coffins, and buried beneath thick layers of sand and gravel. The chambers maintained a steady climate, with minimal daily temperature swings and humidity often below 40%. Occasionally, resins and salt incrustations added another layer of defense against decay. Despite the differences in architecture and burial custom, these sites shared a common advantage: insulation from the outside world. Whether through the thick walls of a pot, the depth of a shaft, or the embrace of desert sand, the tombs of Nuweirat, Saqqara, and Abydos created pockets of stability in a land defined by extremes. It was within these protected spaces that researchers, centuries later, would find the fragile remains of Egypt's first generations, remains that still carried the genetic memory of their origins. Inside the conservation labs of the Egyptian Museum in Cairo, every move is watched, every sample logged. The remains recovered from tombs at Nuweirat, Saqqara, and Abydos are not simply archaeological finds. They are national heritage, guarded by law and by tradition. Before a single fragment could be studied, the Supreme Council of Antiquities reviewed detailed applications from both local and foreign researchers. Every request for sampling, even the removal of a tooth or a sliver of bone, required written authorization. These permits, issued after months of scrutiny, outlined exactly what could be taken, how it would be handled, and where it would travel. Johannes Krauss, from the Max Planck Institute, Verena Schunemann of the University of Zurich, and Salima Ikram, a leading Egyptian bioarchaeologist, stood at the center of this process. Their partnership bridged continents and disciplines, but always under the watchful eye of Egyptian authorities. Ikram, as both scientist and consultant to the Supreme Council, helped navigate the delicate balance between scientific inquiry and cultural stewardship. She ensured that Egyptian voices guided every stage, from the initial sampling to the final publication. The chain of custody for each specimen was unbroken. Inside the museum, Supreme Council of Antiquities officials and curators witnessed the sampling, which was often conducted in climate-controlled rooms, with each extracted tooth root or bone fragment sealed and registered in both museum and ministry ledgers. Material transfer agreements spelled out the terms for any temporary export, down to the number of milligrams allowed and the duration of foreign analysis. Customs declarations, shipping logs, and signed manifests accompanied every package as it left Egypt, with copies filed in both Cairo and the receiving laboratories abroad. The protocols left little room for error or ambiguity. No sample left the country without a clear paper trail, and a promise of return or secure destruction after analysis. The agreements also required that Egyptian scholars be included as co-authors, and that all results undergo pre-publication review by the Ministry of Tourism and Antiquities. These measures, shaped by decades of experience with artifact loss and repatriation claims, reinforced Egypt's sovereignty over its ancient dead. Only after these legal and ethical hurdles were cleared did the scientific team begin their work. The legitimacy of the research rested not just on technical skill, but on a foundation of trust, transparency, and shared authority. In this way, the bones of Egypt's earliest generations remained under the care of their descendants, even as their secrets traveled across oceans to be decoded. Inside the genetics laboratory, the process begins long before a single molecule is sequenced. Researchers suit up in full body protective gear and enter a clean room where the air itself is filtered and monitored for stray DNA. Every surface is wiped down with bleach and ultraviolet light. The stakes are high. A single flake of modern skin or a loose hair could overwhelm the ancient signal they are searching for. The team selects the petrous portion of the temporal bone, the hardest and densest structure in the human skull. This bone, often likened to a natural vault, shields the delicate coils of the inner ear, and crucially locks away genetic material from the ravages of time and environment. Even in Egypt's heat, the petrous bone can preserve up to 50 times more endogenous DNA than a typical rib or femur. With diamond-tipped drills, technicians extract a core of bone powder, sometimes no more than 60 milligrams, careful not to touch or breathe on the sample. The powder is transferred to a sterile tube, labeled, and logged by 2 independent observers. Extraction chemistry comes next. The bone powder is bathed in a series of buffers, first to dissolve proteins, then to break apart cell membranes. Guanidine salts and proteinase K work together to release DNA fragments into solution. Under chaotropic conditions, these fragments bind to silica columns, which are then washed repeatedly to remove inhibitors and contaminants. The DNA is finally eluted in a tiny volume of buffer, often less than 50 microliters. At every step, negative controls, blank samples processed alongside the real ones, are checked for contamination. If even a hint of foreign DNA appears in the controls, the entire batch is discarded and the process starts again. Authentication is relentless. Researchers analyze fragment length distributions, looking for the telltale signature of ancient DNA, short, frayed molecules often just 30 to 60 base pairs long. They check for cytosine deamination at the molecule ends, a chemical scar left by thousands of years underground. Only samples that show these patterns and that pass strict contamination thresholds, less than 1% modern DNA, are approved for sequencing. The result is a dataset built on discipline and precision. Each sequence is more than just a readout of letters. It is a testament to the rigor that shields ancient voices from modern noise. The laboratory's methods, honed over years of trial and error, have finally punched through the climate barrier that once kept Egypt's genetic past out of reach. 90 ancient Egyptian genomes, each one painstakingly recovered from tombs stretching from Upper Egypt to the edge of the Delta, now form the largest genetic snapshot of the Nile's earliest civilizations. In the hands of the research team, these sequences become more than strings of letters. They map out a new geography of ancestry. On a principal component analysis plot, the ancient Egyptians do not cluster with modern Egyptians, nor with sub-Saharan Africans. Instead, their closest genetic neighbors are ancient populations from the Levant and Anatolia. The scatter of data points each representing a single individual from Nuweirat, Saqqara, or Abydos, drifts toward those of Neolithic farmers from the eastern Mediterranean, rather than toward the Sahara or the Horn of Africa. The dataset includes individuals from both the late pre-dynastic and early dynastic periods, covering a window from 3,800 to 2,600 BCE. The genetic signals are consistent across sites and time, confirmed by independent sequencing at the Max Planck Institute, the University of Zurich, and Cairo University. Replication across labs rules out contamination or technical error. The headline result is clear. Egypt's oldest sequenced inhabitants carried a blend of ancestry, about 80% rooted in North Africa, with a substantial 20% tracing back to the ancient Near East. Comparative analyses draw on thousands of ancient genomes from across the Mediterranean, North Africa, and West Asia. The Egyptian samples consistently sit between Moroccan Neolithic farmers and early Levantine populations, with minor but detectable links to Anatolia. No single genome stands out as an outlier. Instead, the pattern is one of gradual blending, a continuum rather than a sharp divide. The data show no trace of a sudden population replacement or the arrival of a foreign elite. Instead, the genetic landscape is shaped by movement and contact, a slow admixture process visible in the subtle shifts of allele frequencies and the shared segments of DNA. These findings lay the groundwork for a new understanding of Egypt's origins. The genetic profiles, visualized on maps and cluster plots, challenge the old boundaries drawn by geography and culture. The story told by the data is one of connection. The Nile Valley, from its earliest days, was linked to the broader world, its people shaped by both local continuity and distant ties. Population geneticists approached the ancient Egyptian genomes with a simple question. How much of their ancestry was local, and how much came from beyond the Nile? Using statistical models like QPADM and admixture, researchers compared the newly sequenced individuals to thousands of ancient and modern DNA samples from across Africa, the Levant, and Anatolia. The results pointed to a consistent pattern. Around 80% of early Egyptian ancestry traces back to North African populations closely related to Neolithic groups from the Western Desert and the Maghreb. The remaining 20% aligns with ancient peoples of the Near East, especially those from the Levant and Anatolia, who had already been farming and building cities for millennia. This blend was not the result of a sudden migration or a foreign conquest. Instead, genetic signals show a gradual mixing that began well before Egypt's first kings unified the land. Admixture dating places the earliest detectable Near Eastern input at least several centuries before the start of the dynastic period, likely during the late pre-dynastic era. Archaeological evidence backs this up. Pottery styles, burial customs, and trade goods from the eastern Mediterranean appear in Nile Valley sites long before the rise of pharaohs. The DNA models fit best when they account for this slow, ongoing contact, rather than a sharp break or replacement. No outliers appear among the sequenced individuals, whether from Upper Egypt's rock-cut tombs or the mastabas of Saqqara. The genomes cluster tightly together, supporting the idea of a shared, blended ancestry across the Nile Valley by the time of state formation. The data contradict the old theory of a foreign elite arriving to impose civilization on a local population. Instead, the genetic evidence supports a story of continuity. A population rooted in North Africa, gradually enriched by connections to neighboring lands. These findings provide a direct answer to one of Egyptology's oldest questions. The builders of Egypt's first tombs and temples were neither isolated natives nor outsiders. They were the product of centuries of movement and exchange, their ancestry a living record of both local tradition and distant contact. The 80% and 20% ratio is not just a number, it is a portrait of a society shaped by both homegrown development and the steady flow of ideas, people, and genes along the ancient world's crossroads. On July 3rd, 2025, the publication of the Nature article sent shockwaves through Egypt and beyond. Headlines declared Egypt's origins upended, its ancient story rewritten by genetic evidence. The findings, over 90 ancient genomes sequenced from some of Africa's oldest burials, sparked immediate debate. Egyptian officials, aware of the study's implications, hesitated to release the full dataset. Within the Ministry of Tourism and Antiquities, concerns grew over how the results might fuel arguments about national identity, heritage, and who gets to claim Egypt's past. Social media erupted. Hashtags like #EgyptianOrigins and #AncientDNA trended across Egypt, North Africa, and diaspora communities. Some voices celebrated the study as evidence of Egypt's historic role as a crossroads between continents. Others worried the data would be used to undermine Egypt's African identity. Afrocentric scholars called for nuance, reminding the public that North African ancestry is inseparable from Africa's broader story, and that genetic mixing does not erase indigenous roots. Nationalist commentators pushed back, warning against letting foreign research dictate how Egypt's history is taught or remembered. Academics responded with both praise and caution. Population geneticists admired the technical achievement, but warned against overreaching claims. Archaeologists stressed the need for more samples from the Nile Delta and Upper Egypt before rewriting established narratives. Editorials in scientific journals emphasized that science is a process, not a final word. Still, the impact was swift. University courses in Europe and North America added the Nature article to their syllabi. Museums in Berlin, London, and Cairo updated displays to reflect Egypt's ancient population as a blend of North African and Near Eastern ancestry. In Egypt, the Ministry of Education convened panels to discuss curriculum changes, though no official revisions followed immediately. The controversy played out in real time. Some Egyptian officials framed the findings as proof of Egypt's openness, not foreign domination. Others urged patience, echoing the study's authors and calling for more data and careful interpretation. International commentators seized on the results, weaving Egypt's story into global narratives about migration, race, and civilization. One detail stood out. After the New Kingdom, sub-Saharan African ancestry increased in Egypt's population. Buried in the technical supplement, this fact became a lightning rod. For some, it affirmed Egypt's enduring African ties. For others, it highlighted how identities shift over centuries. The study's greatest impact landed in the present. The question of what it means to be Egyptian, who inherits the Nile's legacy, remained unsettled, shaped by new evidence and renewed debate. In classrooms, on talk shows, and across digital platforms, Egypt's origins entered a new chapter, written in ancient DNA, and in the voices of a nation still defining itself. Egypt's oldest DNA now proves what textbooks never did. Civilization emerges not from isolation, but from exchange. As ancestry debates intensify worldwide, Egypt's story warns against drawing borders around identity. The evidence is clear. 5,000 years ago, people were already mixing, migrating, adapting. History's roots are tangled by design. What we inherit is not purity, it is possibility. Join the conversation and let us know your thoughts below.